Hello everyone, and welcome to this very special session of the Dutch Mass Spectrometry Society, in which we will be talking on the road towards the Thomson. My name is Esther Thole, I'm a freelance science journalist, and I'll be delighted to be your host for this very special session. And we'll be joined by two guests who hardly need any introduction, I think, in this community, Albert Heck and Ron Heeren. Gentlemen, please join me. Good to see you all in these times of distance, that we can actually be physically together here in the beautiful Koningsbrugge building of Utrecht University. And the reason we're here in Utrecht is not because only of practical reasons that everyone in the Netherlands will understand, but there's also, of course, a strong tie with this university. Albert, you've been a professor here for 22 years. Ron, you've been a long-time professor here.
Correct.
Leaving only, I think, a year ago, finally the ties were cut with your... now fully in Maastricht. We're going to talk about some very special issues, I think. This is not going to be a normal science meeting. We will be talking about career development, about choices you've made, about lucky events, unlucky events, whatever happens when you're building a career like you two have, with, of course, the reason being the Thomson Medal. You both were awarded, and I think it's a very special situation that here in the Netherlands we now have two consecutive awardees. Albert, you got it in 2018.
That's right.
And Ron, you this year.
Yeah.
Still have to receive it, but that will happen at a certain point. So we're going to talk about career development and choices, but before we do that, I think we have to talk about the situation at hand. These are special times for everyone, also in science. Science, as you said before, Ron, to me, where we were talking before this take, it is a group effort, it's a social endeavor. So how are you coping and how are your groups coping with doing science in times of a pandemic, with all the restrictions? I mean, doing mass spectrometry from home it's kind of difficult. So Albert, to start with you, how are you coping? How is your team coping?
Yeah, I think you all need to be fair with this. It's impacting all of us, and not in an only positive way, mostly in a negative way. But you also see that the strength of an international science team of people makes it also very interesting to cope with these pandemics. We had in our group a girl from Wuhan who already in January came back from China. So we actually knew about the COVID virus already very soon, and luckily she was not infected, but she came back with a small cough, and so it started already at that time. But more now, it's tough. We are back in the lab, but only for 30%. And you also see that, the great thing, what I see, all these international people in the group, for them it's also really difficult. They are isolated from their families. They may have families in countries that are affected. Those parents or so or friends are worried about them. They don't have a big network of friends here.
Especially when they just arrived here, it must be incredibly lonely.
Yes, and what I see then as the power of the group, and I think that's maybe for more people, but certainly for scientists, that people really care about each other. And we work hard, but we also care about each other. And there are all kind of initiatives where they do things with each other, do things for each other, and talk with each other. So socially, I think it's now very important to stick together. Yeah, work-wise, I think we also have to be fair. We are not working on 100% of our capacity, and certainly, and I discussed this already with Ron a bit, you can still do practical work, but really what you need in science, these creative discussions, that is way more tough in these periods of time.
Yeah, those are hard to replace by a Zoom meeting.
Yeah, and I must also say I'm very happy that we discovered Zoom and Teams and WebEx and whatever. That's an eye-opener, and that's one of the few positive things about this whole crisis. But it doesn't replace this face-to-face meetings that also the NVMS wanted to have here for this occasion as well.
Yeah, it would be a lot nicer with an audience. Yes, it would have been better. And you, how are things in Maastricht?
Very similar. Also operating at lower capacity, also about 30%. We are now cranking up our practical education again, which is a challenge because that means if we have students in the lab, our researchers can't be in the lab because of the restrictions. But the most important thing that we notice is the lack of social interactions. And while we all work hard, we used to play hard, and that's now very, very difficult to do. I mean, social interactions, going out, discussing over a meal the results of the day, and just making fun of each other and of me.
Celebrating the normal things you have in the group, like thesis defense or...
Well, last week I had one of my students defend this thesis all online, and this was my first online thesis event, and it just felt weird. It felt like I was in one of these Zoom meetings, and I appreciate what you're saying, that those are great tools that we use and also enrich our lives, but are they suited for these celebratory moments? I have my doubts. No. And it's a shame, that the person cannot celebrate that success at the end of 4 years hard work and lots of papers and lots of discussions.
Yeah, and looking forward to that. And then it's a bit
bleak. And also not for these people. And we also had a thesis defense and, the proud parents, which is always a highlight if they are there as well. Yeah. They could not travel from Spain to this defense. So, we all look back at our PhD defenses and other things in life that you want to celebrate. Yeah, and then you really feel a pity that they cannot do this at the moment. And we can only hope that soon this will go back a little bit to normal.
Yeah, let's hope for that. Yeah.
So one of the upsides, and that's kind of interesting thing what happened, since we're all working at home in the lab, or not in the lab, but working at home, there's a lot of backlog of lots of data that we've gathered in the past is now all being processed. So I see a lot of productivity in people finishing papers where they normally would not get to finishing papers, lots of theses being read, so they're also upside, but that will dry up at some point.
If I, now that you mentioned upsides, and then you think again, what a great field we are in. Because if you talk about mass spectrometry, you're not generally think about COVID.
No.
But if you now look what's happening in research, in diagnostics, in tests, maybe we are biased, but you see mass spec coming back in the tests, you see mass spec coming back in the fundamental research to discover the structure of the virus. So you also, in all these bad things that are happening, you again see some pride in that your colleagues and yourself are involved in also this problem, scientific problem. So
that your preferred technology is actually being
used for good purposes.
Yes. You almost think that mass spectrometry can contribute to everything, and now it's evidence again that
it's true.
In COVID, it's for sure true.
Did you any of you actually hop on the COVID research bandwagon? Is a bit too negative, but is there any COVID research going on, or did you decide, well, we've got nothing to contribute here, let's focus on the other things we're doing?
We did a little bit of work. One of my colleagues, Peter Peters, with his electron microscope, actually made pictures of the coronavirus in organoids together with Hans Klevers from Utrecht here and Marion Koopmans. And we did a few molecular pathology studies on pathological tissues from COVID patients. But it's not going to become a research line. So we're not going to hop on the bandwagon. We helped some other people with their research. But other than that, it's not going to be a research line.
Maybe I can add. I think you phrase it right, hop on the bandwagon, because I also looked it up. I think there are now more than 30,000 papers about COVID already published. We also run a European facility, and so we also get people that come to us to execute their research. And so in that sense, we have now samples from Bologna area in Italy, from doctors there that approached us. And in those samples that we have of about 60 patients at 3 stages of the disease, we're trying to see what the antibody responses are in these people. And so I think that is, I find it hard to say no to such a project.
Mm-hmm. Exactly.
But I don't know, you don't do this anymore for thinking that you get a big breakthrough, but you really hope that you can help if it was only in getting better treatments for the people. And I must say, I'm also very proud of that, because when you get these samples, you actually think about the history of these samples. And as I said, we have a couple dozen patient materials. Yeah, and a third of them, they are not among us anymore. And that gives a totally different perspective. And I must also say, it's also been good for the group because our lab was totally locked down for a few weeks or months, except when you did COVID-related work. And what I actually saw, that several people in the lab volunteered and asked, oh, can I please be involved? Maybe because they wanted to leave their homes, but I also actually think because they really want to contribute to this research.
You can really benefit now and show the benefit of investing, I think, in science and in infrastructure.
Yes,
for unexpected events.
Yes, yes. And I think that's certainly for the mass spec infrastructures that are around, not only Ron's and mine, but all over the world. You see that happening.
Yeah, I think you're absolutely right. It also shows that we need to keep on investing in science and scientific infrastructure to be capable as a society to respond to these type of pandemics. And if we lose our investments in science, then it will become much harder to start that up again and to actually make a difference in society.
Yeah, you'll lose valuable time. Talking about investing in science and infrastructure, I think, Ron, you made a small movie of things going on in the lab, so we can see some actual footage of what is going on, so we can all see it from our homes. Shall we have a look and you talk us through what we're actually looking at here?
Absolutely, it's a good idea. Good. So what we're looking at here is actually our use of a detector from CERN, the TimePix detector, where we're using that for intact protein imaging and intact protein detection at the end of a time-of-flight mass spectrometer. All the little blobs that you just saw were individual ions, and every color of the blob was actually an individual mass. So in a single image, we make this time-of-flight instrument actually into 252,000 detectors. And the beauty of that is that we can now use that to very sensitively, with one single ion, examine surfaces, mixtures, look at proteins and protein complexes. And actually this detector is one of the detectors that Albert and I are using on a joint project as well. So what I like about this, and I sometimes name it from CERN to the surgeon, because the detector came from CERN, and we're now using it to actually look at pathological samples that we're getting out of the OR in Maastricht. So,
and samples from
patients,
from
patients, biopsies, resected tumor materials,
or other tissues, or
resected tumor materials we do a lot of, and then we make very thin sections, we fire a laser at it, and then we shoot these proteins out of those tissues, trying to determine what a tumor boundary is, or how the profile of the different tumors we find predicts how a patient will respond to therapy. So there are a lot of things you can use this technology, but why I like this particular detector so much, because it's really going from one spectrum at a time per laser shot to 262,000 spectra at the same time. And the concept of going to parallel detection in mass spectrometry, that's something that we're only scratching the surface. And those are the things that that are to come in mass spec, and where I think will really push mass spectrometry forward.
And this is really new, what we've just seen
This
here.
is brand new. This actually was recorded 3 days ago. It's the latest version of the TimePix detector that actually has a higher detection frequency, which means we can get, if you think about a movie, we can get more frames per second. In the past, we were able to get 100 frames per second. Now we're able to get 1,000 frames per second, which means we can collect even more spectra and even more pixels and get more information in our data sets. At the same time, that's a huge challenge because all that data is in there, but you also have to process it and get the data out. So that's one of the challenges that we're facing.
But that hasn't changed, I think, over the decades.
As
technology
goes forward, and as we improve the technologies in our mass spectrometers that we work with, we will also have to improve the ways we use our computers, algorithms to correlate things. And that's of all ages. It's essentially Moore's law for mass spectrometry. You could define that.
Yeah. Finally, on this note of what is going on right now, you already mentioned, Albert, the use that you see of mass spectrometry in COVID research. But is there anything else going on in the field, let's say over this past year, that stood out for you? Either developments in mass spectrometry technology or in applications, or a paper that really was a surprise to you, a pleasant surprise, I mean.
Yeah, that's always difficult to pick your favorite child. Pick your favorite paper.
We won't hold it against you, whatever you pick.
No, but I think...
Pick imaging mass spec now.
Yeah, but I think what is sort of nice, you know, if you get a bit older, you get more and more asked, what do you think is going to happen in the next 5 years? And I find so great about mass spectrometry that my answer is always the same. If you asked me that 5 years ago, I would be totally wrong. And that's because the technology is still so much alive that, you know, we are ready for the unexpected. But if you really press me, you know, I think what Ron said, getting more resolution, getting more information on a tissue. Another drive is to go to more sensitive measurements. There are a lot of people doing proteomics, but they want to do now single cell proteomics, so I find that a nice development. Our own involvement is more where we actually want to use mass spectrometry as a single particle detection analyzer. So instead of the 256,000, we want to focus on the single ion at a time. And we and some others have just shown that this is possible on the Orbitraps, but only if you take them apart and modify them a bit like we do all the time. And this opens up all kind of new avenues to really study large particles, which I also like, study viruses, gene delivery vectors, etc. And so I find it hard to pick in mass spectrometry. There's so much exciting going on. Mm-hmm. And I find it is also interesting in our relationship. We have our own niches, but we always try to find each other. And at that crossing, we also find new things to do. And like in Ron's project, where he wants to do the imaging of the intact proteins, you started with smaller molecules, lipids, etc. And I keep bugging him also a bit from why not look at proteins? And we developed some technologies with new fragmentation methods, introducing lasers to do top-down proteomics. And now you see that they can come together, and in essence, both are not new technologies, but if you bring them together, you can make the next step again. And that makes this field so vibrant and for me so exciting.
Actually, to add to that, I think that's also one of the strengths of mass spectrometry, because we are capable as mass spectrometrists to act at the interface of disciplines, and whether it's different technologies coming together or whether you bring together people with a medical background with fundamental molecular scientists. And mass spectrometry is always at that interface while still being a very vibrant research field by itself. And I think that's also what makes it so exciting, and what also gives, at least me, but I think you're very similar in that way, the drive to actually move forward. Because if you bring these things together, that's when you really make a difference with your research.
Yeah, no, I fully agree, and that we indeed share. You never ask what's possible, but you sort of ask what do you really want to do, and if you don't know how to do it, then just start doing it. And I think that's sort of what brings you further and what brings us further. We sort of, you have your great role models in science, but it's not good to follow them too much. No. It's also, and sometimes I even say to my people, don't read all these papers, just do what you think you should do. And then you sometimes come up with really innovative things. And when a technology is at your core, like mass spectrometry, it gives you that freedom to make such moves, and I think that's so great about this field.
Yeah, absolutely.
Well, it's clear that you're still very much fascinated both by mass spectrometry, long careers, and I think it's time to move on to that main topic of why we're here today: career development and building a career in mass spec, as you both have and still are doing, and as I understand you both right, you're still planning on doing a lot more here. There are quite some parallels between the two of you. You all both alluded to that as well. You're about the same age. Both did your PhD in Amsterdam. We briefly before talked about where you first met, and apparently it has something to do with an NVMS meeting probably, which I think is a very nice coincidence, of course. You also both were lucky to get, maybe lucky is not the right word, but we'll get to that, a full professorship at quite an early stage in your career. I think you were 34, you were 37. That's quite young. Is that, to start off, is that also a bit of a blockage? I mean, many people many young researchers strive for a professorship, but when you have it already that early in your career, is it then hard to stay motivated? Or not at all? Is it just more motivational to keep on going? How did that happen? I want to get to that, I think. I mean, how did you get to that stage? Was it lucky with the technology? Was there a big drive? Was it just, were you on the right moment that, riding the wave, the field was increasing and there was investments? What was it? Or was it a deliberate choice? Did you always want to become a professor?
For me, yes.
Yes, you did?
My mom once told me that already at 4 years of age, I told her I was going to be a professor.
Sure.
When I was tinkering with radios, or not at 4, but a little bit later on, she repeatedly said that. So I don't remember that, I will be honest, but she has told me that, so I believe her, of course.
Yeah, of course.
But it was a little bit of all of that, I think. But it was also a little bit of a deliberate choice in my career. Because I knew that once you're at that professorial level, also you get new possibilities to lift your research at yet another higher level. And so it's definitely not a blockage. I would say it's almost an instrument. And yeah, we've been very lucky that we got that at a relatively early age. And ultimately, what it allows you to do is just to follow your passion and do it in a way that holds you back less. Mm-hmm. In the sense that if you're a postdoc, you always have to run things by your professor. Or once you're the professor, you sort of get to drive and direct your own research ideas.
The independence.
Exactly.
But how do you get there?
For me,
yeah, well, I mean, I think many of the younger people in the audience watching this want to know, how do you get there? Because it's not easy.
But it's and there's also no recipe.
True. But we like to hear yours at least.
My story is quite funny. So before I became professor in Utrecht, I was lecturer at Warwick University. Mm-hmm. And different from Ron, I never said that I wanted to be professor, and I also think I didn't think that. But I wanted to do research, and I didn't want someone else to tell me what research to do. So basically, that means that you maybe want to be professor, but that's not how I did see it. If they gave me money, a lab, and I could do what I wanted to do, I think I was happy. And that's maybe also at that lecturer level and postdoc level, you're really in the lab and you can do these things. But maybe to tell the story, I was actually, Utrecht was looking to fill a professorship in mass spectrometry. I didn't apply because, first of all, I didn't know about it. And secondly, at the age of 32, if I knew about it, I probably also would not have applied. But they actually had a famous mass spectrometrist, Fred McLafferty, in the search committee. And so I vividly remember I was just looking at some spectra in my office in England, and I got this call from the States. And Fred McLafferty has a very specific voice. I cannot repeat that here, but basically he only said, "Oh, but you have to apply." And I said, "To what?" and so he was really instrumental, I think, on both sides, pushing me to apply. And I tried to explain him, Fred, this is America, this is the Netherlands, they only appoint people that are 50 plus, etc. And I think he also convinced people here in Utrecht, he's a young guy, but at least invite him as well and let him tell his story. And I had to go here, and I don't know all the details, but there were like 8 applicants and we all had to give an interview. And for me, it was more a fun thing. I thought, they will never hire me. And
you had nothing to lose.
I had nothing to do. And I was actually pretty happy in England as well, I have to admit. That was a lab where I worked together with Alexander Makarov and with some other people in the mass spectrometry field. So and that's the other part of the story. So he called me on Friday. And this is really true. On Saturday, I was going to sign a contract to buy a house in the UK. And if he hadn't called on Friday, but if he had called me on Monday, I might have signed that contract and I might have said to Fred McLafferty, sorry, I just bought a house. I will not apply.
I'm staying here.
So I tell this story because I find it funny, but also, that's not building your career and looking for your road ahead. You have to be a bit lucky. When I came here for the interview, I was really motivated to show my best. And I saw it as an opportunity, but I didn't have the plan, oh, I will be hired here. And maybe last thing, which, it's nice to be appointed at that age, but I sometimes also tell, you almost get from postdoc to professor instantly, and that's also quite a difficult transition in the beginning.
It has to be difficult, yeah.
Yes, but it comes back to what I said before, if you don't know how to do it, just start doing it and see where you go, and don't care that you sometimes make mistakes as long as you try to do your best.
I think one of the most important things in life is luck to some extent.
So we could say in part, of course, you got the position in Utrecht because of what you brought to the table and the trust you could, of course, inspire in the committee, but it was also really a lucky break that someone else got your attention, definitely. Yeah. How was that for you, Ron? The same lucky breaks, or did you do it all on your own?
A little bit of both, but luck is a big factor. Being at the right time at the right place, knowing the right people that contact you. For me, it's a relatively similar story. When I was a young student doing my bachelor and doing an internship, after doing my bachelor, I thought, what these PhD students can do, I can do that too. So I went to the director of the institute, of the FOM Institute. I said to Frans, I think I should be a PhD here. I think I can do this. And he said, oh, Mr. Heeren. So you think that's, well, you know what? You go to the study for this particular exam at the UVA, a very difficult quantum electrodynamics exam. And if you pass, you can start as a PhD student. So I did, and I started immediately after my bachelor's as a PhD. So the interesting one, that was a lucky break. Mm-hmm. And then I had a great...
Well, not only a lucky break, I mean, you took the initiative at least. Yep. So it goes to show that if you at least try.
Yes, you have to take the initiative. Yeah. I mean, don't think for somebody else. That's what I even tell my own children. If you want something, you go for it and you try for it. And that's what I did at the time. And then after that, doing the PhD, I knew I was doing a PhD in nuclear fusion science. Cool. So physics. And then I thought, this is not the field where I want to be in, because that was changing so rapidly, or that was not changing rapidly enough. And at that time, when is nuclear fusion going to work? In 1992 was 60 years from now. I think if you ask the fusion scientists the same question now, it's still 60 years. Yeah. So that was a good choice. So
you wanted to move to something else. And how? Mass spec.
Mass spec. So I was working on hydrogen, on protons, and I wanted to move to proteins. And there was a group at AMOF at the time that was doing biological mass spectrometry, the group of Jaap Boon and Piet Kistenmaker. And I already knew them from my interactions at the coffee corner at AMOF. And at some point they needed somebody to build a mass spectrometer, build an ICR. And I said, hey, I already built a mass spectrometer for fusion research. Why don't I build one here? Again, the opportunity arose, I took it. And within 2 years, I was the person doing instrumentation development at FOM together with my colleagues. And from there, it all of a sudden accelerated. And the big change for me when I thought, okay, what else can we do with these mass spectrometers? Mm-hmm. And already at that time, we were doing laser-based technology. I thought, I really want to do something with imaging. I was always fascinated by photography and the technology behind it. And then we wrote a very big proposal to actually do imaging mass spec at AMOF and developing the technology for that. And from there on, that became my main topic. And then another lucky break 25 years later was when a surgeon from the Maastricht University Hospital visited me, Steven Olderdaming. He's a very good friend, a person that I collaborate with closely. He was doing experiments with me, and we were sitting together behind this instrument, the mass spectrometer at FOM. And at some point he said, we should do this in a clinical environment. And I told him, that's actually why we do this. But at AMOF, in this very high-tech environment, making that transition or translation from...
It's a completely different world.
I mean, even the AMC, which was the AMC, the Academic Medical Center, was 5-minute, 10-minute bike ride away. We could not get a solid project started. And Steven immediately came up with lots of ideas, and he said, there is an opportunity for us to get the province to invest in new institutes. What would you need? I said, so many million we would need. And he said, I think that's not impossible. I said, well, if you manage to do that, I'm in. And so it happened. We wrote the proposal, we got funded, and we were asked by the board of directors of Maastricht University to build this institute some 6 years ago.
That's the M4I.
The M4I, the Maastricht Multimodal Molecular Imaging Institute.
Who thought of that name?
Yeah, me. But the cool thing is, I think M4I, Maastricht for Imaging. There you go. So that's the brand now.
Yeah. Very
so
cool.
in being at the right time in the right places, meeting the right people, accidentally, serendipitously, played a big role.
But what I find, what we also have in common, and what I hear now again, you're even more than me. You have a physicist background.
Yep.
I had a discussion also with my son, who had to choose which subjects to do at high school, and he didn't do biology. And I said, "Oh, why not biology?" And he said, "Yeah, you also didn't do biology," which is true. So at high school, I didn't do biology. I went to study chemistry in Amsterdam, and I disliked the biochemistry courses a lot. I did physical chemistry, I did theoretical chemistry. I thought it was way fun. And at that moment, I was not attracted to biology at all. So if you talk about careers, what I find sometimes strange is that someone studies analytical chemistry, they say, oh, it's an analytical chemist for the rest of their life. Now, certainly, Ron is still a little bit of a physicist, but not a true physicist anymore. Yeah, and I also don't know what I am anymore. I also started a bit as a physical chemist, but now I'm more a biologist or a biochemist, and I think that's also part of luck or taking opportunities. And it's not that we went into biology because there was money, but maybe something changed in my mind that I thought. First, I thought first I thought biology was very vague. And then I thought, hey, it's so vague, there's so much to discover. And
we develop. I think so. The technology developed, but I think we develop in the way we think.
Yeah, but I think it shows that you should not stick to your field too much. Be open.
Unless you really like it, I can imagine. If you're really happy in what you do.
Yeah, but then you should still be open for things that can come from another field that may help your field.
Yeah.
I always told myself I will do something different every 5 years, and so far throughout my career, I've been able to do so.
Well, that depends a bit, of course, on the perspective that you take. If you look at it from a cosmic perspective.
I've been doing the same thing. But still from a perspective as a scientist looking at, even at mass spectrometry, the different applications, it can be small things, but if you stop challenging yourself, you also will not progress anymore. Yeah. So, and I think
But were there ever moments that you doubted your choice? Critical points that you thought, this is not going the way I want, or this is not what I thought it would be, or maybe I should consider doing something completely different?
It's coming back to what we discussed before, and maybe Ron has it as well. I became professor very young. And then at some point, and I don't know when this was exactly, maybe when I was 45 or so, you're going to think, okay, I'm going to be this 20 more years. I've been this already 10 years. And then I cannot call it a crisis, but I really thought, I don't know if this is really what I want. And for me, luckily, I had the opportunity then to do some sabbaticals at the ETH and at the Scripps Institute. And actually what I sort of discovered, that my job was really fun. Mm-hmm. So instead of stepping out, I sort of stepped in again. But I think it's also very logic that even if you like your job very much, which I do, there are times that you also think, okay, is this it for life? What are the other opportunities? And when you're really young, when you're 20, I also had that feeling that I still could become an astronaut or a football player or whatever. And of course, that at a later age, you realize that these changes are not that dramatic anymore. But so I don't say I have had my doubts, but more that you really start to rethink, okay, how I'm going to keep this exciting for the rest of my life. And now I have, no doubt, and I never regretted staying in Utrecht. Utrecht is a very nice place for me. But you sometimes also think, oh, what would have happened if I had gone to the States or somewhere else?
I will tell you that I moved 6 years ago to Maastricht. Retrospectively, I've told a few people I should have done this 10 years ago because it completely changed my thinking being in a medical environment. And I always tell people I started as a physicist, I became a professor in chemistry here at Utrecht, and then became a professor in the Faculty of Health, Medicine and Life Sciences in Maastricht. So by making that step and making that change, also after a sabbatical, actually, it's kind of interesting, so sabbaticals help. it really made me think differently about myself and the way I perceive it. Having said that, though, there are always ups and downs.
Of course.
I mean, the position where we are now, we also have to deal with lots of people in administration, directors, and board of directors.
A lot of tasks that feel like chores, basically.
Yes,
You have to do them, but...
And I realized this Corona crisis, we started about that. As a result of it, I'm much more in the lab now, working with some of the students, maybe as an excuse to be in the lab, could be. But
we won't tell anyone.
I think I just did. but I notice how much fun I have just really doing a very simple experiment. And it's just fun. That's how we started. I'm not going to say that I'm going to do it
in
the
24/7.
students
Still allow you to be in the lab? Because I also know from some professors that their students rather not have them mess with their equipment anymore.
If he says yes, then I move to Maastricht.
Yes, you're allowed. Of course, we would love to have you to come to Maastricht, Albert. I'm very well supervised by my students.
Oh, good. Well, that's reassuring.
Me too. They keep me in my office.
But coming back to what you just said about, well, thinking about your move to Maastricht, and maybe I should have done this earlier. When you look, both of you, when you look back now, with all you know now, and go back to the start of your career, would you go for the same choices? And be honest.
Yeah, that assumes that you made choices, and that
you always make choices. They might seem
They-
unconscious, but they are choices.
my answer would be that I don't know if it would be mass spectrometry, but what I want to say again, as a mass spectrometrist, you're like the spider in the web. You're in the middle. You can reach out to biologists, you can reach out to physicists, you can reach out to medical researchers. So I really, and I think that's what we share as well, we are sort of technology nerds. And from this technology basis, you can really reach out to, and I learn every day from my collaborators. That's also something what I say more often. And that comes about by being an expert in this technology. And if you say, would you do it again? Now, if I know what I know now, I would say yes, this was the perfect moment to go into mass spectrometry. If I had to start again, I would maybe choose mass spectrometry again, or another technology that you think, hey, with this technology, I don't have to be limited to a certain area. I can really reach out to other areas as well, because that, for me, made it really, makes it really fun.
And go for an academic career again also. Or
yeah, if I don't become prof footballer and play, and then
then another prof. Yeah.
No, yeah, I think, I find it a difficult one. We also collaborate a lot with people in industry, pharmaceutical industry. Sometimes you wonder how would have been, as I said, a good friend of both of us, Alexander Makarov, we were postdocs together in Warwick. Yeah, he went into industry, became extremely successful there. Sometimes you think, okay, that would also, I don't say that I would achieve what he did achieve, but you would like sort of to know what would have happened if. Yeah. Yeah. But, that's also nice that you don't know the answer.
And Ron?
I also find that it's a question of conscience, right?
It is.
Because on the one hand, I would say I would do exactly the same what I did now, but I know that I wouldn't, for the very simple reason that circumstances will change going back. What I would do the same, and I think that's very much in line with what Albert said, I would choose a technology that sort of has connective capabilities, that's capable to cross different domains, because that's something that I would like. What I would also not do differently, keep on looking for opportunities to challenge myself. Whether I would end up in mass spec or different technology, I don't know. That might change. But the type of choices that I made, I would not change. At some point, I was offered, also because I'm sure to a position in industry, where I actually visited and explored, and I've explored other positions also in academia, and said no. So you're right, you make choices. I've never regretted my choices. So in that sense, I would do the same thing again, but it would be completely different.
And what about we hear a lot about the high pressure that we face in academia. And there's a lot of going on. Younger people are voicing themselves, well, we don't want this anymore. You're from the generation before that. I mean, to get to where you both got, it takes a lot of hard work. Do you consider yourself to be a workaholic?
Oh, I ask my children, they'll say yes. And I think I am a workaholic, but it's not work. And I realize it is work. Don't get me wrong, I'm not stupid.
You're not in denial.
I'm not in denial, no. But it's so much fun that and there are also parts that, like you said, they're chores and you have to do.
Of course, but that's in every job.
That's in every job. But it is so much fun to interact with all these young people. And I realize I'm in a very easy position to say this because, as you said, we're there where we really want to be and where we drove ourselves into a position that we wanted to. But for the young people that we work with, they have an even harder task ahead and a more daunting task ahead because the pressure from the outside world is actually building. And I think the good news is that within the Dutch academic system, we're trying to look differently at recognizing and rewarding these young students and trying to change that because there is somehow the expectation that our assistant and associate professors have to excel in everything. If you honestly think about that, that's impossible.
Yeah, of course.
So work with people's talents and make them excel from there, and don't only judge them on the things that they are not capable of. My anecdote in that always is I'm a volleyball trainer, and at some point I have a volleyball training license, and I had to do a positive coaching training to keep my license. And what I learned there is if you work with your players in a team and you only make them do what do good already and make them even better in that, they're going to be so much more productive because they will enjoy what they're doing. Next season, I tried that with the team I was coaching at the time. We came out on number 2. And all of the ladies told me, this was the best season ever. It was so much fun. What did you change? And actually, the only thing I did, I was not trying to improve what they couldn't do, but I was working what they could do. And so maybe in academia, we should maybe try something similar.
Do you agree?
No, it's a very...
First the workaholic part. We don't get over that. Do you consider yourself a workaholic? And do you need to be one to get to be successful?
I don't, this is a really honest answer. I don't think I work hard. And still I work almost all the time. I'm busy with my work.
Yeah.
And that's our perception. So if you ask me, do you find yourself a workaholic? It's the same as Ron. The answer is no. If you ask people around me, they will say yes, and maybe also the people in the group. What I find difficult, and Ron said that already, pressure on all of us is very different and changes every decade. From the times that it took me 3 weeks to type an article and then send it by post to a journal to how it is now.
Wow.
Yeah, all these things have changed, and there's a lot of pressure, not only on the young people, also on us to adopt to these systems. I'm totally fascinated by all the things we study, so I always think about it. But it's still finding a balance, and it's also having a family who wants to support it. So that makes it all. It's already very enjoyable. But if your family doesn't back what you're doing, then you also have troubles. So then it's sustainable and it's doable and it's also fun. If you need to be a workaholic, I don't know. I find that always a nice experience. Also in our lab, we have researchers, women who become pregnant. I actually sometimes see that they become even more efficient and in their time management, in how they do. I almost get jealous and I think, oh, maybe I should also be more efficient in how I do things.
It's also a phase in life where you're in, because I look at it differently now, and my kids that are now grown up look at me differently and the time that I spent with them than they did when they were 6 or 7. Yeah. And then I was in the house much more time a day, and my hours that I made were also a lot less. So you adapt.
I don't know. I think I made long hours.
all the time. But science is, of course, I think we can all agree that if you don't work hard.
Yeah, but I think
if you're passionate about it, it doesn't feel like working hard. So do we make a lot of hours? Undoubtedly. Yeah. And I think Albert and I both.
Yeah, but that should not be the standard thing to do.
Yeah,
do you enjoy what you do?
Yeah,
basically?
But I think
if
you're.
Feel like.
Yeah, and it's sort of the same. If you're curiosity driven, you cannot switch that off.
No, no way.
And so each time, so I run a lot, and if I run in the forest, I'm thinking about work. and not about the stressful parts of work, but
No, but the creative part.
Yeah. And I can't switch that off, and actually I enjoy it. The running is easier if you have to.
I do the same thing
Something
on the bike.
to think about.
I had a discussion with one of my students recently, and Ron, you know, it's amazing. When we have a work discussion, all the topics you ask questions about, how do you know so much? I was thinking, I don't know that, but it's exactly because I'm curious and I want to know, and I don't know the answer that I asked the question. And maybe that's something what, you know, if you're that passionate and that curious, you're always busy. Somebody's telling, how would that work? And then maybe the both of us are extrovert enough to throw it out there quickly.
Yeah. Other point, the Thomson Medal. I think widely regarded as the most prestigious prize in mass spectrometry.
True.
How important is such a prize? What does it do? What does it mean?
What does it mean? I think for me, and I've said this in an interview at some point, for me it's really the best part of the Thomson Medal is the fact that it's a recognition from all of the mass spec societies across the globe. And that means that I must have done something right, and that the choices that we were talking about earlier are appreciated by other people. So that's a very important element for me. And everybody likes to be recognized for what they do.
Yeah.
And so the pat on the back.
It's on the top of the Maslow pyramid.
Exactly, yes, exactly. So from that perspective, that makes it perfect. But at the same time, when you say that, you also have to realize it's not about me. You do this with a huge team of people, with organizations that support you, with friends that you collaborate with.
That sounds very sympathetic, but in the end, you get the medal.
True, which is why, different story. But I think if you don't share that recognition and you don't make very clear in everything you state about a medal like that it's really a team effort. Yes. We are at the forefront and at the face of receiving that medal, but the recognition goes to everybody. For me personally, it also, if I look at the list of people who got the medal in the past, and Albert being preceding me, so he's on my list now.
Following the footsteps.
Those are my mass spec heroes that are on there, and people that some of them I still interact with, I actually do research with, and those are the people that I really hold high in our field. And being able to, you know, somebody telling you that you're amongst those, that is an amazing feeling.
Yeah, no, I can add to that. You know, it's also the name Thomson. That's also so great about our technology, that we can say, oh, we are talking about all these innovations. And this technology started in 1898. You know, so it's the history. I think also with the Thomson Medal, it's given at this conference that we hope to organize in Maastricht in 2022, where the whole community comes together. And you know, it's also part of the community building that there is an hour of the conference where these prizes are given to the leaders of the field. Now, and before I got the Thomson Medal, that was already for me the highlight of the meeting, that you got a lecture of a well-respected person and you got a medal, or she got a medal. And so when you look at that through your young career, and then at some point some people decide to give you that award, and you think, whoa, I didn't expect to be there, but they probably want me to be there now, and you feel very proud. And I share with Ron, you feel proud and you know that you didn't do this, but yeah, you also did do this. You know, I'm proud of that.
You don't get the award for nothing.
No, but you're proud that you generated a group around you with whom you could start this creative process to get where you are, and that you get the recognition. That you know, that is nice for you, and you should, I think, also fully enjoy it. But you know, I think it's these prizes are part of our community. And I think we should just cherish it, and I hope, you know, that's what you also hope, that in 100 years from now there are still Thomson medals given to people in mass spectrometry, and that gives it all so much more stature than just this medal. And so, you know, I start to I must say I really appreciated this recognition from the whole mass spectrometry field.
From the whole community.
Yeah, you know, it's even more. I've had some other awards, but this is really the people who can judge you best, and
They
so
actually understand what you do.
no, I don't want to say that the others don't, but
Oh, you
if
can.
someone does understand what you do, then it's of course your fellow mass spectrometrists,
Yeah.
and so in that sense, it's very much appreciated.
I want to get back to that idea of community and being among friends, but I think first it would be good to take a look at the research again, because there are more clips we want to show, and I think we're now going to see one of you, Albert, from your lab. So if you can talk us through, and then we'll get back to the research and the community and club. So, Albert, we're going to have a look at your lab. What are we seeing here?
Yeah, so in our lab, what we typically do is we buy commercial instruments, and I say it's like buying a commercial car. But the first thing we do is we open up the car, take the hood off, and then we're going to modify or pimp up the car or pimp up the instrument. What you see here is we are investing a lot in lasers. We have like 3-4 lasers here, excimer lasers, IR lasers. And we use these now to come to a new sort of sequencing of peptides, but even more so sequencing of proteins and protein complexes. So you see the lasers going in at the back end of a commercial Orbitrap mass spectrometer. We really opened this up, we made a hole in it, and we are ready to go to shoot these lasers to get sequencing information about proteins and protein structures.
And is this something really new? Is this something that has been developed over the past few months, years? Can you tell me a bit more about?
I think so. This development of using lasers for photodissociation is already older. But the way we do it now, and especially that we do it now on whole viruses or whole antibodies, is something that we are really pioneering in the world. And we are learning it also the hard way. So it's really a team of people that do this. And the first time we did shoot the back hole of the instrument, and it died. So you learn these things by falling and getting up again. But now we are really getting nice data. But this doesn't happen overnight. It's really an investment of a couple of years of work.
And what do you expect this to do? You said we can study whole viruses.
What we are aiming to do, and this is really aimed for the future, we like to find which antibodies are in someone's plasma against pathogens like COVID. And we think we can sequence these antibodies, but we think that we need partly this equipment for it to do it. We think a lot of people are trying to do this, and we think there's still a missing link. And by studying these antibodies with lasers and also with electron capture dissociation, we think we can fill that gap, and we would be one of the first to be able to really de novo sequence these important biomolecules, these antibodies that protect us against pathogens.
Yeah, so that is a strong research line for the near future.
It's a very strong research line. I find it very, we have like a team of 8 people working on this in the group, all with different disciplines. Some have expertise in lasers, some in bioinformatics, some in clinical samples. And they all work together to achieve this. And yeah, it has been a team effort. And now is about the time, I hope this year, next year, that we will get our big breakthrough.
And interesting maybe to add to that, it's also something that we're going to work on together in the future.
Aha.
Absolutely.
Yeah.
Absolutely.
In what way?
Well, actually, what Albert was just showing, the photodissociation and looking at the structure of proteins and protein complexes, the detector image that I was showing earlier on, what we're trying to do is actually merge the two together in order to add a different level of information of structure of protein complexes by using the setup that Albert was just showing to dissociate and blow apart all the different subunits of these protein complexes such that they fly apart. And then we image all of the different complexes in a small time of flight and know the stoichiometry at the back end of an Orbitrap. So we can do the studies that Albert is doing, the sequencing and the structural analysis based on the components, but we can also start to image, which is where we come in, the structure of the protein complex. So it's a little bit, if we succeed, and it's a very challenging and daunting project, but we'll be competing
Otherwise
with electron
you wouldn't
microscopy.
be doing it, I think.
Of course.
And what is nice, I think we will never succeed in this project if we would do it alone. So
No.
it's really where
Impossible.
the expertise of the two groups comes really together, and that's also quite unique in the world. So I don't know if we will make it, but it's the road towards it is already
It's a
very
lot of
nice.
fun. Yeah.
Oh, you'll definitely make something, I would say.
Yeah.
Yeah. So nice plans ahead for the future. Plenty of hopeful, interesting news coming up for the Dutch mass spectrometry community.
Absolutely.
Yeah, very good. We'll be looking forward to that. We're almost at the end of this session, but before we leave the audience, I would like you both to give some advice to the younger researchers watching this. We've talked about career development, we've talked about choices, lucky breaks, daring to change subjects, keep challenging yourself. What would, very short, but what would your advice be? It can also be don't listen to other people, could be, but I think you have something to share. Ron.
So my advice to the young mass spectrometers that are now pursuing a career in mass spectrometry would be bold, challenge your peers, challenge your professors, but most importantly, follow the passion that you have for your topic. As much as these professors seem very confident, they also don't know everything. So it is perfectly fine to challenge them. But at the same time, keep your drive up and dare to be different. Dare to challenge the world around you, because that ultimately is going to help you forward.
Well, clear advice for those daredevils out there. Albert?
Yeah, what can I add to that? No, I think you should also really be in an absorption mode. We are getting so much information and so many possibilities. If you work in a lab or if you study something, don't get that wasted. So really absorb. And I see sometimes people who think, yeah, but what this lecture today, that's not really directly related to my subject. Should I listen? And I say, yes, please do, because maybe not now, maybe not tomorrow, but in 5 years' time, you will think, hey, I've heard that before. And that's, I think, what has helped me really always, also go to maybe symposia, go to lectures where you think, this is not my topic, but just see what you pick up there. And one day it will come back to you. And I think it's almost like with kids, and I think as a young mass spectrometrist, you're a kid in mass spectrometry. Be open to everything. We were at the time when I did my PhD, electrospray was just born, and the first years I didn't know about it. And it's not that people directly tell you, here's the technology, here it will be, this will change your life. But I remember that I first went to these lectures and I thought, I don't see much use to this, and it's maybe not so interesting. But you go and then you get attracted to something like that. And I think that's this open-mindedness, and be ready for the unexpected, and always try to look for something that you don't know yet. Can bring you very far, maybe not directly tomorrow, but it can bring you very far.
Yeah.
And maybe to add that, because you raise a very important point, and that's also educate yourself by going to conferences, etc. So maybe to the young people in the audience today, IMSC 2022 will be in Maastricht, and all of your young NVMS people should actually come and educate themselves. What do you think?
Yeah, no, and be sure that everyone knows that you have been there. So go to this PI that you admire and say, here's my poster, I want to show it to you, I want to discuss this with you. This is how you start your network. And actually, maybe now I'm an older guy, if a student comes to me with that question, I'm honored. Because I say, oh, this person finds it important that I come to their poster. So please reach out and don't be scared to approach anyone in the field. They are all friends, they're all happy and nice people, and so keep interacting in our community.
Is it a friendly community?
I know also other communities, and this is a very friendly community, which doesn't mean that we now and then compete.
We compete all the time.
But you can be friends because you're also friends.
Yeah.
You can be friends and compete.
Absolutely.
Or is the level of competition, how is that? Is that very, it's not a very cutthroat field, I would say, or?
I think it also depends on the country where you do mass spectrometry. I think here in the Netherlands we're very lucky, because while we do compete for funding and for, let's say, positions at universities with our colleagues, it's never as cutthroat as sometimes you find in the US or in the UK even.
Yeah, but I compare it also with sports. If there was no competition, there would never be world records anymore.
Yeah.
Exactly.
And that's here as well. We continuously challenge each other. You know, we also in a positive and negative way. If Ron tells something exciting what he has been achieved, I say, oh, but you can't do this yet. Now, and that's always my answer, I believe.
Back to the drawing board. But then I tell
Nothing
him,
personal,
just
nothing
wait
personal.
and see, Albert.
Yeah, no, but you need that challenge as well.
Yeah.
And so competition is good as long as you do it open and
As you have a good idea of why the
professional
competition
way.
is there.
Yes.
Not a personal thing, but it's about
No.
the content, the science,
Yeah.
the field, the technology.
And the community. I mean, the NVMS is one aspect of this big international community. So for the young people there, realize that Mass Spec is an international endeavor and is a very international community. So you can explore the world while following your passion. So, but you can only do that if you are part and actively part of that community.
Yeah. So to sum up, you should think broad, be open, be bold, challenge yourself, and don't be afraid to approach other people.
Exactly.
Yeah?
Exactly. Yeah.
Perfect.
Good summary.
Well, thank you. Well, thank you both very much for taking the time to sharing your ideas about career, about everything else in science, and coming all this way to Utrecht, Albert. Really appreciate that. No, Ron, Albert, thank you very much. I hope you all enjoyed watching this, and of course, for any questions, both professors are very easy to interact with, and even right after this session, there will be a live Q&A with Albert and Ron, where you can ask all the questions you may have. For now, thank you very much for watching, and as Ron said, hope to see you all in Maastricht in 2 years from now. Thank you.